glance at some of the work that we're doing to try to deliver to breeders tools that they can use to improve root systems, not as an end in itself, but as a way to boost yield and increase resistance to abiotic stresses. First project I want to highlight is a project funded by the International Wheat Yield Partnership to develop this toolbox for breeders. It's a consortium of a number of different groups that have been independently chasing QTLs for different root traits. Roberto and Silvio's group at Bologna, John Innes' group, University of Queensland, UC Liebig University, Judith, Simmet, and ourselves, and some private sector breeders are involved as well. And the aim of the project really is to then introgress these QTLs into leaf germplasm so that breeders can test them in the field. And, fortunately, most of the loci that all of the different groups are looking at are independent, so we're not looking at the same gene. So two of the outputs from this project are, first of all, the germplasm, these new nils that have different expression of root traits, and also then the markers for these root traits. And there's two important aspects of the validation. So the first is the question that was raised earlier. If you're doing the selections and the QTL discovering on seminal roots or seedling root traits, do they are these phenotypes expressed in mature root systems in the field? If you look at seminal root traits at root angle, for instance, does that also carry through to crown root angle? And then also, what is the effect on root distribution at depth? And then the other question, of course, is does that really matter? Does it have an effect on yield in the field situation? So the first one that Roberto already introduced is seminal root angle, very easy to screen, and you've got, in this particular case, our students working on a double haploid mapping population of the parental lines that contrast for root angle in winter wheat. And she's used this clear pot method, just a pretty high throughput method of imaging the roots in this clear pot and then deriving the root angle. And she managed to find the QTL on 4D. It's the only QTL that was actually consistent across 3 sets of experiments. Lots of QTLs, or several QTLs that were uncovered in each individual experiment, but there was only this one in our hands that was consistent. Unfortunately, the D genome of the wheat doesn't have very good marker coverage, and so the QTL region is quite large. So she's working with deletion lines then to narrow this down, and there's one line that has a smaller interval that covers the QTL region. Just incidentally, this doesn't have necessarily anything to do with deep rooting, but since we've been talking about it a bit today, these parental lines also show differences in the ability to form the rhizosheath, and that could be quite interesting to map as well, so she's working on that. It could be that part of the explanation for the differences in the rhizosheath formation is due to the genotypic differences in the secretome, or the compounds that are secreted by the roots. And this is one particular polysaccharide that shows quite a bit of variation in the tails of this population for the rhizosheath. Then lots of tools you can use then to look at the material in the field. So we heard about the Chevolomics way of very quickly being able to measure crown root angle. But ultimately, you really want to look at the deeper roots, and we saw there's lots of different ways to do that. There's the hard way of doing it, digging a trench against the profile, trench profile face method. The advantage, though, is you get to see roots as they are in the field situation, but from the genetics and breeding point of view, we need throughput. So so you can use these kinds of rhizotrons, again, limited in the throughput. Another indirect way that we've been working on is using a DNA-based method. So you can extract the DNA from a sample of soil, use, in this case, a wheat root-specific primer, and then relate it to the root mass in that sample. And it works fairly well. So the DNA-based method is are the triangles, and you can pick out differences between two genotypes like this. So it's semi-quantitative, and it still needs a bit more work. It's not necessarily the cheapest method either. But the other important thing is that because it's specific to the species, and you can develop another primer, for instance, for a weed, blackgrass, the very important weeds in the UK, and you can determine the relative proportions of the two different weed root species in a soil sample. But it doesn't tell you anything necessarily about the actual root mass. It's an indirect method. Another indirect method that we've used that's mentioned before is looking at root activity rather than the actual physical presence of roots. And one way to do this is to measure patterns of soil water extraction. So installing these PVC access tubes and then using the Centec soil moisture meter, making sequential measurements of soil moisture every 10 centimeters over the course of a season. And in a drought experiment, you can show this data. This is old data now, but it shows that over 1.2 meters from the soil surface, some varieties are able to extract more water in these deeper soil layers than another variety, this Xi19 variety, better than spark, and it's also more drought tolerant than spark. So that makes sense. So it's faster than excavating the roots, but it's an indirect method, and it's still really not the throughput we would need to do a whole mapping population. So another method that we've been trying to develop that would be an indirect way of looking at changes in soil moisture content and then inferring from that the presence and activity of roots is electromagnetic induction. So it's a commercially available kit that farmers use to map their fields for soil conductivity, but you can use it in a way that you can get fine differences in root moisture content. And this shows that over a period of soil drying in the field, you can detect differences between different wheat genotypes using this method. The barrier we have now is actually to do the inversions from the soil apparent soil conductivity you get from the instrument to an actual soil moisture profile is computationally quite demanding. So we need someone to work with us on developing the software and computing to be able to use this in a routine way. And then finally, so lots of examples of coring, and there's really no way to get around that. And Anton has developed methods that are at the point now of throughput where we can do some genetics in the field. And just an example of what we did in this Wheelby project that Roberto introduced, looking at soil root distribution to the soil profile of different tillage practices. Again, not much happening, or not many differences at depth, but in the superficial layers, it seems to be that shallow non-inversion tillage promotes root growth compared with conventional plow. And then finally, just leave you with one of my favorite quotes from Weaver that I think is appropriate for our discussions today, and a silly film. Thanks.